Today we are going to talk about the digestive system. So first, what is digestion? Digestion is the chemical and mechanical breakdown of food, and it's the breakdown of food from larger substances into smaller substances. Those smaller substances will be absorbed in the small intestine. The food we eat includes carbohydrates, lipids, nucleic acids, and proteins. These foods are polymers, or you may have heard them called biological molecules. These foods need to be broken down into their smallest subunits called monomers in order to be absorbed in the small intestine. Carbohydrates are broken down into monosaccharides or simple sugars. Lipids are broken down into glycerol and fatty acids. Nucleic acids are broken down into nucleotides, and proteins are broken down into amino acids. It's only the monomers that can be absorbed into either the blood or the lymph. Carbohydrates, nucleic acids, and proteins are absorbed through enteric cells of the small intestine into blood capillaries, while lipids are absorbed through enteric cells into lymph fluid, specifically into lacteals, which are specialized lymph capillaries. There are two parts to the digestive system. There's the GI tract or alimentary canal, and there's the accessory digestive organs. Let's take a closer look at these two parts of the digestive system. The GI tract or alimentary canal is a passageway from the mouth to anus that is considered an external environment because both the mouth and anus are open to the external environment outside your body. We're going to see that the alimentary canal functions in digesting food and then absorbing those fragments, or digesting polymers, breaking them down into their smaller fragments called monomers. The parts of the GI tract are the mouth, pharynx, esophagus, stomach, small intestine, and large intestine. The accessory digestive organs are either going to help the GI tract digest food by mechanically tearing it apart or providing excretions, whether it's digestive juices or enzymes that aid the digestive processes. The accessory digestive organs are the teeth, tongue, and gallbladder. The digestive glands are the salivary glands, liver, and pancreas. Let's trace the route of a piece of food as it would travel from the mouth to the anus. So first, food would be ingested into the oral cavity. From the oral cavity, it would then flow into the pharynx or throat. It would flow first into the oropharynx and then the laryngopharynx. From the laryngopharynx, food would then enter into the esophagus. The esophagus connects the throat to the stomach. From the stomach, food would then flow into the small intestine. The first part of the small intestine is the duodenum, followed by the jejunum and then the ileum. The duodenum is the shortest part of the small intestine. The jejunum is the next largest part, and the ileum is even longer. The ileum leads us into the large intestine. The first part of the large intestine is the cecum. The next part, the ascending colon. Next, transverse colon. Then descending colon. Next, sigmoid colon. Finally, the anal canal, which includes the anus. There are 6 essential activities for digestion. These are ingestion, propulsion, mechanical breakdown, digestion, also called chemical digestion, absorption, and defecation. Ingestion is taking in food to the oral cavity. It includes processes such as chewing, also known as mastication. Propulsion is moving food from point A to point B. Swallowing is one such process, so is peristalsis. Both swallowing and peristalsis are muscle contractions that move food from place to place. Peristalsis is a smooth muscle contraction common in organs like the esophagus. Mechanical breakdown includes chewing that occurs during ingestion, and mechanical breakdown is going to be tearing or physically separating large polymers into smaller segments. We're also going to include mixing and segmentation. Segmentation, like peristalsis, is also a smooth muscle contraction that moves food, but not place to place. Instead, it's going to move food against the walls of the GI tract in order to bash or churn it into smaller pieces. Digestion here is talking about chemical digestion, wherein enzymes and other substances are able to perform catabolic reactions or are able to break down larger substances into smaller subunits. Absorption primarily happens in the small intestine. Nutrients that have been broken down into their monomers during the process from the beginning to the end of the GI tract at this point would be absorbed into the blood or lymph. Defecation is an elimination step, is elimination of indigestible foodstuffs and waste. This is a good image to study to look at these different parts. We can see that ingestion occurs in the oral cavity and then proceeds through the other steps such as mechanical breakdown, propulsion, absorption, chemical digestion, and defecation. Other structures we want to take a look at are things like the salivary glands. These are an accessory organ. There are 3 salivary glands. They produce saliva. The salivary glands are the sublingual, submandibular, and parotid glands. Other accessory glands we haven't looked at yet in any detail are the liver, the gallbladder. Both the liver and the gallbladder have ducts that go to the duodenum, which is the shortest part of the small intestine, also the first part of the small intestine. The pancreas also has ducts that go to the small intestine, specifically, again, the duodenum. So more information on those digestive processes. Ingestion, you can know that as eating. Propulsion, as moving food through the digestive tract or GI tract. Mechanical digestion is for increasing the surface area of molecules by tearing them apart. This increased surface area provides more space for enzymes to act and break down these larger subunits even further. Chemical digestion occurs when bonds are broken, so they are catabolic reactions. And usually chemical digestion is going to finish the process of breaking down polymers into their monomers. Absorption is a transport process, transport through the enteric cells of the small intestine into either the blood or lymphatic vessels. Lacteals are specialized lymphatic capillaries that absorb fat or the breakdown products of fat, such as fatty acids and glycerol, whereas the blood is going to absorb the monomers of carbohydrates, proteins, and nucleic acids. Defecation is the elimination of feces, which includes indigestible foodstuffs and digestive waste. Here are 2 smooth muscle contractions. Peristalsis is a part of the propulsion step of digestion. And we can see here that the back and forth contraction and relaxation in adjacent segments of the GI tract moves food from one area to another. Segmentation is also a smooth muscle contraction, but it belongs to the mechanical digestion process of digestion. And segmentation is going to have concurrent or contractions that occur at the same time in order to mix or move food back and forth. And this mixing and churning is going to be a way to further process or break down food mechanically. It wouldn't hurt to review the abdominal regions and quadrants that you see in Unit 1 of your lab manual. Being familiar with the anatomy of the abdominal pelvic cavity will help in the understanding of the digestive system. Another structure we want to talk about is the peritoneum. The peritoneum is a serous membrane that surrounds the abdominal pelvic organs, specifically the digestive organs. The visceral peritoneum covers the surfaces of the digestive organs, whereas the parietal peritoneum lines the walls of the abdominal cavity. In certain areas, you'll see a double layer of peritoneum. This double layer of peritoneum is called a mesentery. Organs that lie within the peritoneum or within a mesentery are considered intraperitoneal or peritoneal organs. If they lie outside of or behind the mesentery, they are called retroperitoneal. Here are some examples of mesenteries. Again, mesenteries are double layers of peritoneum. The greater omentum is a blanket-like mesentery that lays over most of the digestive organs. Here it's lifted up. We can see the proper mesentery that connects most of the jejunum and ileum of the small intestines together, and the mesocolon and sigmoid colon, which anchor those parts of the colon to the abdominal cavity. There's even the lesser omentum, which is a mesentery that connects the stomach to the liver. You also need to know the histology of the digestive tract. First and foremost are that there are 4 layers or 4 tunics to the digestive tract. The innermost layer closest to the lumen is the mucosa, followed by the submucosa, muscularis externa, and then the serosa. So these layers are written from inside to out, or they're written as close to the lumen to further from the lumen. The mucosa consists of an epithelial sheet with underlying connective tissue. In some mucosas, there's also a muscularis mucosa, or a small band of smooth muscle. The submucosa consists of connective tissue that contains blood vessels, lymphatic vessels, and nerves, and also some glandular tissue that can produce mucus such as serum mucus and other chemicals. The muscularis externa contains 2 layers of smooth muscle. The 2 layers of smooth muscle include the circular and longitudinal layers, meaning that the fibers of these layers are perpendicular to each other. These are going to be the muscles that contract to perform functions such as peristalsis and segmentation. The serosa is the outermost layer of the alimentary canal wall, and it's actually just the visceral peritoneum. It contains fibrous connective tissue, and in organs that are not completely surrounded by the visceral peritoneum, they also have an adventitia. For example, the esophagus is not found in the abdominal pelvic cavity, and so it's going to have an adventitia on its outside instead of a serosa. There's also nerve plexi. There is a myenteric nerve plexus and a submucosal nerve plexus. These are nerve fibers from the sympathetic and parasympathetic nervous system, and they are going to allow the GI tract to respond to nervous system stimulation. Here's just another image showing you that the inside layer or tunic of the GI tract is the mucosa, then followed by the submucosa, then the muscularis externa, and then finally the serosa. Serosa is only found within the abdominal pelvic cavity. So here you're actually looking at the most inferior part of the esophagus, and while most of the esophagus has an adventitia at its outermost layer because it's in the thoracic cavity, this lower part below the diaphragm, this part of the esophagus actually does have a serosa, but it's only a small portion of the esophagus. We're going to look at some basic anatomy for the rest of the GI structures for the GI tract, or what we're calling the alimentary canal. So the mouth is also known as your oral cavity. It's going to be bounded or surrounded by your cheeks laterally and then anteriorly by your lips. The tissue that lines the inside of your oral cavity is stratified squamous epithelium. We see stratified squamous epithelium also in the oropharynx, the laryngopharynx, and the first part, or primarily large portion of the esophagus. So here we're seeing structures of the oral cavity, such as the uvula. The uvula is skeletal muscle that is able to move up or superiorly to block the nasopharynx when we swallow. This keeps food from going up into our nasal cavity or out our nose, and instead it's supposed to go into our esophagus after leaving our throat. We can see the superior labial frenulum and the inferior labial frenulum, which are pieces of tissue that connect the lips to the gums, and there's even a piece of tissue called the lingual frenulum that connects the tongue to the base of the oral cavity. The tongue is also made up of skeletal muscle. Here is a better view of the uvula, and we can see how its contraction could pull it upwards and block the nasal cavity, therefore making food go down into the oro and then laryngopharynx, and then into the esophagus. The esophagus is a skeletal muscle tube that is behind the larynx and trachea. So esophagus is posterior to your windpipe. Another structure we're looking at just briefly is the tongue. It's skeletal muscle. It contains bumps called papillae. The papillae, again, are bumps on your tongues. In all of them except filiform papillae, the bumps contain taste buds. You have 3 salivary glands: a parotid gland, a submandibular gland, and a sublingual gland. These glands have ducts that lead to the oral cavity. The purpose of saliva is to keep both the mouth and the food we eat moist. There is an enzyme called salivary amylase that begins the digestion of starch in the mouth. There's also secretions that neutralize acids and dissolve food chemicals. We need saliva in order to use the chemoreceptors that are on our tongue. So saliva has a couple of different functions. It contains both enzymes, buffer systems, and mucus. But it also contains a high amount of water. The parotid gland is going to be in front of your ear. The submandibular gland is going to be under the mandible, and the sublingual gland is under the tongue. Let's briefly talk about the pharynx. The pharynx is your throat. and it's a cavity both behind your mouth and behind your larynx. It actually has 3 parts, but the parts that are involved in the digestive system are just the oropharynx and laryngopharynx. And this is where food is routed before it's routed into the esophagus. The tissue that we find in the mucosa of the pharynx is also stratified squamous epithelium for the oropharynx and laryngopharynx. The esophagus is also a long muscular tube. It's much longer than the pharynx, and it's going to be able to use the muscles in its muscularis externa to perform peristalsis, which is contraction of adjacent segments that move food from point A to B. Here, point B is going to be the stomach. The esophagus will send food from the pharynx to the stomach. If we looked at its gross anatomy, we can see that on one end there are pharyngeal constrictors, or constrictors that are going to persist or function in swallowing, and those are going to be in the pharynx. On the other end of the esophagus, there's the cardiac sphincter, also known as the gastroesophageal sphincter, and when the sphincter relaxes, it allows food to leave the esophagus and enter the stomach. The esophagus is going to have stratified squamous epithelium as its epithelium in its mucosa. And when there is no food in the esophagus, it folds in on itself and its mucosa appears to be highly folded. So here we can see the folded mucosa of the esophagus. It has an adventitia instead of a serosa. And then if we look at the esophagus to stomach junction, we can see a change from stratified squamous epithelium to simple columnar. So as we move from the esophagus to the stomach, there will be a change in the mucosal epithelial lining from stratified squamous to simple columnar. So far we've looked at the mucosa of the oral cavity, the oropharynx, the laryngopharynx, and now the esophagus. All the structures that we've looked at so far had stratified squamous as their mucosal epithelial sheet. When we move into the stomach, we'll be looking at a different mucosa. This mucosa is mainly simple columnar with gastric pits. So the stomach is a pouch that's going to be bounded by sphincter muscles. The sphincter that is going to attach the esophagus to the stomach is the gastroesophageal sphincter or cardiac sphincter. On the other end, the pyloric sphincter leads from the stomach to the duodenum, or the first part of the small intestine. We're looking at the gross anatomy for the stomach. It's positioned under the diaphragm. It's in front of the pancreas, and it's going to be medial to the spleen and also a little in front of the spleen. It has a rough J shape. It has structures that we'll look at, which are the cardiac sphincter, cardiac region, fundus, body, pyloric antrum, pyloric canal, and pyloric sphincter. We also want to look at the fact that the stomach itself has 3 smooth muscle layers. It has an oblique layer, whereas most GI tract organs just have a longitudinal and circular layer in their muscularis externa. The inside of the stomach has many folds in its mucosa, and these folds are called rugae. If the stomach is empty, the rugae are visible, but it can stretch to hold up to 4 liters of food at a time. So here is the anatomy of the stomach. The area where the esophagus meets the stomach is called the cardia. This would be where the cardiac sphincter is located, also called the gastroesophageal sphincter. The area above where the esophagus meets the stomach, this domed region is called the fundus. The main portion of the stomach is called the body, where we have narrowing at the base of the stomach near the pyloric sphincter is the pyloric antrum. We have even further narrowing to the pyloric canal, and that leads us to the pylorus. This narrowed region of the stomach past the body is called the pyloric region. And then, not pictured here, but the pyloric sphincter would connect the stomach to the small intestine, specifically the first part of the small intestine called the duodenum. The lesser curvature is right here, and it's going to be closer to the liver, whereas the greater curvature is going to be right here. It's much larger than the lesser, and it's closer to the spleen. Here's an image that shows the oblique layer of the muscularis externa that we see for the stomach. So again, the stomach is special in that it has 3 layers in its muscularis externa, a longitudinal, circular, and oblique. We can see the rugae of this emptied stomach. Those are folds of the mucosa. We have a better view of the cardia, the fundus, the body, the pyloric antrum, pyloric canal, and the pyloric sphincter, which are going to be near that pyloric part of the stomach. And then here, the pyloric sphincter would lead into the duodenum, that first part of the small intestine. If we were to look at the mucosa of the stomach, we would see that the simple columnar epithelium is dotted with many gastric pits. These gastric pits include enteroendocrine cells that release hormones, but also cells that produce substances like a bicarbonate buffered mucus that protects the simple columnar epithelium from digesting itself. Because one of the other secretions that the stomach can produce is hydrochloric acid. This hydrochloric acid produces a very acidic environment inside the stomach, and this acid is produced by parietal cells found in these gastric pits. This acidic environment is perfect for an enzyme called pepsinogen to become activated into pepsin. Pepsinogen is produced by chief cells, and in the environment of the stomach, which is very acidic, has a pH of about 2, pepsinogen is converted into pepsin. Pepsin is an enzyme that breaks down protein. So pepsin is very important for protein digestion, and protein digestion actually begins in the stomach because of this enzyme pepsin. So parietal cells produce hydrochloric acid, chief cells produce pepsinogen, and then pepsinogen is converted into pepsin in the stomach because of the acidic environment that is present. The small intestine has 3 parts. It is made up of the duodenum, jejunum, and ileum. The duodenum is the first part and the shortest part of the small intestine. Food is mainly absorbed in the small intestine, and we'll see that things like bile and pancreatic juice are both dumped into the first part of the small intestine, the duodenum, to aid in digestion. If we were to look at the basic anatomy, the small intestine goes from the pyloric sphincter of the stomach all the way to the ileocecal valve that would lead food into the large intestine. We look at the accessory organs that are going to empty fluid, whether it be bile salts, pancreatic juice, that's bicarbonate, or enzymes. They're going to empty these secretions into the duodenum. The duodenum is the first part of the small intestine, and we can see that the pancreatic duct and the common bile duct both empty into the duodenum at the hepatopancreatic ampulla and sphincter. That is an area that allows fluids to enter the duodenum via or through the major duodenal papilla, which is a bump that we find on the inside of the duodenum. The gallbladder has a cystic duct that merges with the common hepatic duct to form the common bile duct. The pancreatic duct is not as complex, but it's going to bring secretions from the pancreas, whereas the common bile duct is bringing secretions from both the liver and the gallbladder, even though it's primarily the gallbladder. The function of the liver is detoxifying. It also is able to make bile, and bile is not stored in the liver, instead it's stored in the gallbladder. So it's important to know that the liver makes bile, but the gallbladder stores it. So hence there are ducts that connect the liver to the gallbladder. All right, if we were to look at the microscopic anatomy of the small intestine, we would see modification to its inside layers. There are circular folds, villi, and microvilli in the small intestine. These structures reside or are a part of the small intestine. Circular folds are permanent folds in the submucosa and mucosa of the small intestine, and they allow food to spiral throughout the small intestine, but also increase the surface area for absorption. Villi are going to be permanent folds and finger-like projections of the mucosa. These finger-like projections are going to be made up of simple columnar epithelial cells, and what's inside each villi is a capillary, a blood capillary, and a lacteal. On top of the individual simple columnar epithelial cells are projections of the plasma membrane. These projections of plasma membrane also appear finger-like and are called microvilli. So circular folds are permanent folds of the submucosa and mucosa. Villi are going to be projections of that mucosa, and then microvilli are projections on the cells. Altogether, we have many projections and folds and divots that we find inside the small intestine, and this greatly increases the surface area for absorption in the small intestine, which is the main function of the small intestine. Here we can see the circular folds that have permanently folded parts of the mucosa and submucosa. And we could see how spiraling of the food is going to progress through the small intestine. If we look at one of these mucosal folds, we would see that there are projections at the top. The projections that are closest to the lumen here are called villi, and what's lining the villi are small cells. These are enteric cells. They are simple columnar cells. They're the cells that we have in the small intestine. If we were to look at just one of these cells, we would see that their plasma membrane also has projections. These projections are microvilli. So microvilli are located on cells. Villi are located on the mucosa, and circular folds are permanent folds of the submucosa, including the mucosa. Lacteals are inside, along with blood vessels, each villi. And what we're seeing here are folds upon folds upon folds that increase the surface area for absorption in the small intestine. Next, we're going to move on to the large intestine, and this is an area where some water absorption takes place, but it's mainly going to be an area where only water or electrolyte absorption occurs. Most of the function here is to dehydrate any leftover indigestible waste. The gross anatomy, if we look at it, we can see multiple structures. The structures we're going to look at for the large intestine are the colon, which includes the ascending, transverse, descending, and sigmoid colon, the rectum or anal canal, the anus, the appendix, and the cecum. Those are the structures of the large intestine. So here they are in order. So as food moves from the small intestine to the large intestine, it would enter first the cecum, which is a pouch just after the ileum. To get to the cecum, food would have to pass through the ileocecal valve. From the cecum, food would then move into the colon. It would first move into the ascending colon, and food would move up. Then into the transverse colon, food would move across. Then into the descending colon, food would move down, and then into the sigmoid colon, and that is an S-shaped region of the colon. From the sigmoid colon, food would move into the rectum, and then through the anal canal, then out of the body. There are some unique features of the large intestine: the Tinea coli, haustra, and epiploic appendages. Epiploic appendages are just fat-filled pouches that we find in the visceral peritoneum that surrounds the large intestine. Haustra are pocket-like sacs that are caused by tone of the Tinea coli. And the Tinea coli are longitudinal smooth muscle that we find in the muscularis externa of the large intestine. Haustra are going to participate in strong segmental contractions that occur in the colon to move food from point A to point B. Food is actually considered feces by the time it moves into the large intestine. So as it moves from the ileum to the cecum, the food is now officially feces. So let's just trace the route of food through the large intestine. This is the last part of the small intestine, the ileum. So to move from the ileum or small intestine to the large intestine, we pass through the ileocecal valve into the cecum. From the cecum, food moves up the ascending colon, then right at the colic or hepatic flexure. It's going to move then through the transverse colon, then down past the left colic or splenic flexure. It'll move down in the descending colon, then through the S-shaped sigmoid colon, finally through the rectum, then the anal canal. The anal canal includes an external anal sphincter, and the internal anal sphincter is a part of the pelvic floor. Another structure we haven't mentioned is the appendix, sometimes called the variform appendix. The variform appendix is located in the large intestine, but instead has an immune function. We're going to move on to looking at our accessory glands. There is the liver, which is the largest gland. It has plenty of functions. The main ones you need to be concerned with are its production of bile. It makes bile. And it's going to also be able to detoxify substances, whether that be blood or lymph. It's able to respond to hormones and drugs. It also makes some of our blood proteins. The bile that it makes is stored in the gallbladder, and we'll see that bile is going to be useful for breaking down fat in a process called emulsification. The liver has 4 lobes, a right and left, a caudate lobe, and a quadrate lobe. We'll see that the cells of the liver are called hepatocytes, and these hepatocytes form plates that look like hexagons with a central vein. On each corner of these hexagons, there is a porta hepatis. The porta hepatis contains a blood vessel, both an artery and a vein, and bile caliculi. Here is the right lobe of the liver, left lobe of the liver, separated by the round ligament. The gallbladder is actually under the liver. If we look at the back of the liver, we can see the caudate lobe and then the quadrate lobe of the liver. So the left and right lobe of the liver are much bigger than the other 2 lobes. And again, the gallbladder right here, we can see its close association with the liver and how it just appears to be a sac-like pouch. Here's a better image of the liver and the gallbladder so we can see the duct system. The left and right hepatic duct become the common bile duct, which joins with the cystic duct from the gallbladder to form the common bile duct, which leads to the duodenum. Pancreatic duct leads to the duodenum, so secretions from the gallbladder, liver, and pancreas can enter that first part of the small intestine. Next, we're going to look at the microscopic anatomy of the liver. We already talked about the name for liver cells, that is hepatocytes. Hepatocytes are hexagonal in shape, and they have a central vein. On each point of the hexagon, there is a portal triad or portal hepatis, which includes an arterial, venual, and bile duct. The bile duct are continuous with the bile caliculi that go into the hepatocytes. And these are structures that help generate or produce the bile that will then be sent to the gallbladder for storage. The gallbladder is a muscular sac. We saw it in the last couple of slides. It stores the bile that the liver produces. It is attached via the cystic duct to the common bile duct, and through there it's able to secrete bile through that duct into the duodenum. The pancreas has both exocrine and endocrine functions. It's important to know that it has both, and also that the endocrine cells are called pancreatic islets, and the exocrine cells of the pancreas are called acinar cells or acinar cells. So just more anatomy on the pancreas and the gallbladder. This view here may give you an idea as to the orientation of the pancreas. Its head is going to be surrounded by the first part of the small intestine called the duodenum. Its tail is going to abut the spleen. The gallbladder is going to be under the liver but above the duodenum, and its cystic duct combines with the common hepatic duct to bring bile to the duodenum, just like the pancreatic duct will bring digestive juices and enzymes to the duodenum as well to help aid in the breakdown of polymers to their monomers. That way that absorption can happen in the small intestine. The exocrine glands in the pancreas secrete digestive juices such as bicarbonate ions, and we find that the secretory cells for the exocrine glands of the pancreas are localized in the acinar cells. The endocrine gland cells that produce hormones such as insulin and glucagon for the pancreas are localized in the islets called the pancreatic islets or islets of Langerhans. Again, the pancreas has both endocrine and exocrine functions. Next, we're just going to take a closer look at some of the things that we've talked about thus far. Another reminder here is mesenteries are double-layered pieces of peritoneum. Some of the mesenteries we looked at were the lesser omentum that connects the liver to the lesser curvature of the stomach, the greater omentum that has a lot of fat in it and it overlies most of the abdominal pelvic cavity, mesentery proper, which connects parts of the small intestine, specifically the jejunum and ileum, the transverse mesocolon and sigmoid mesocolon that are going to attach those parts of the mesocolon to not only other mesenteries, but to the abdominal pelvic wall. Here's just another view of some of those mesenteries we talked about. Here is the greater omentum, not pulled superiorly, so we can see how it blankets or overlies the abdominal pelvic cavity and all the fat pouches that are found therein. So it is a site for fat storage and also is going to hold some arteries and other vascular structures in place. And here's the lesser omentum, and we can see how it attaches the stomach or suspends the stomach to or against the liver. Another look at the proper mesentery that we find between parts of the small intestine and the mesocolons that anchor those parts of the colon. Again, mesenteries are double layers of peritoneum. Next, let's look at those steps of digestion again, and let's talk a little bit more about those enzymes, about emulsification, and about absorption. So we know that chemical digestion is a catabolic process. We're breaking down large substances into small substances or macromolecules into monomers. We need them to be small enough for absorption, and most absorption takes place in the small intestine. The chemical digestion includes enzymes. These enzymes are intrinsically made and dumped into the gastrointestinal tract. Enzymes are able to break down food if it's already been broken down via mechanical digestion as well. So that step of digestion is also important. Without water or the mucus that's produced and contains water in it, the GI tract would not be able to utilize these enzymes to break down the macromolecules into monomers. We'll focus first on the digestion of carbohydrates. Carbohydrates are our polymer that we want to break down into a monomer. The monomer for carbohydrates is a monosaccharide or simple sugar. Examples of monosaccharides include glucose, fructose, and galactose. The carbohydrate digestive enzymes are listed here. If you see the word amylase, go ahead and assume that it's a carbohydrate breakdown enzyme. For example, salivary amylase, pancreatic amylase, but there's other enzymes that don't end with amylase that breaks down carbohydrates. A lot of them sound like the disaccharides they break down. For example, lactase breaks down lactose, maltase breaks down maltose, and sucrase breaks down sucrose. Sucrose, lactose, and maltose are disaccharides or double sugars that have already been broken down from polysaccharides or even bigger sugars. So the process of carbohydrate breakdown will break down large polymers or polysaccharides into disaccharides and then finally monosaccharides. I highly recommend these study images. Here you can see that carbohydrate digestion begins in the mouth with salivary amylase, continues in the small intestine when pancreatic amylase is dumped from the pancreas into the duodenum. At this point, we will use both pancreatic amylase and brush border enzymes to break down lactose, maltose, and sucrose into galactose, glucose, and fructose. We'll break down disaccharides to monosaccharides. So carbohydrate digestion begins in the mouth and ends in the small intestine. Protein digestion begins in the stomach with pepsin. Pepsin is converted from pepsinogen in an acidic environment to pepsin, and pepsin is a great protein enzyme. Proteases are a common term for an enzyme that breaks down protein. Pancreatic proteases include trypsin, chymotrypsin, and carboxypeptidase. Trypsin is able to activate chymotrypsin and carboxypeptidase once they're secreted from the pancreas into the small intestine. Brush border enzymes are aminopeptidase, carboxypeptidase, and dipeptidase. Again, peptidases break down protein. The brush border is another term for the microvilli of the enteric cells in the small intestine. So these enzymes hang out on the microvilli of those enteric cells, hence their name, brush border enzymes. Protein digestion begins in the stomach with pepsin, ends in the small intestine with our brush border enzymes. We'll see that proteins are broken down into large polypeptides, but not as large as full proteins, then to even smaller polypeptides, and finally into their monomer amino acids. The digestion of lipids involves a pre-treatment state called emulsification. Pre-treatment using bile salts is emulsification. Bile salts are going to be able to break down fat globules, and if we can break down fat globules, it's easier for pancreatic lipases to act on those fat globules. will then break down the lipid into fatty acids and monoglycerides. So here is emulsification. Again, emulsification is the pre-treatment step that fats must go through in order to be absorbed through the enteric cells into a lacteal. So here's a large fat globule. It's really hard for lipases, enzymes that break down fat, to act on a large substance like this. So bile salts are released from the gallbladder into the small intestine to act on fat globules and break them down into smaller fat globules. These are much smaller and easier to act on, so the enzyme lipase is able to break down these fat globules coated with bile salts into monoglycerides and fatty acids. The remaining bile salts will then coat the broken down fat and create micelles. Then a recombination of the fatty acids, monoglycerides, and bile salts will create chylomicrons. Chylomicrons are formed after the micelles have passed through the enteric cells of the small intestine, and they are what are absorbed through or into the lacteal. The lacteal, again, is a specialized capillary, and here we're seeing fat absorbed into a lymphatic vessel. All other polymers, once broken down, are absorbed into blood capillaries. So we can see that the breakdown of fat begins in the mouth with lingual lipase and gastric lipase, but that's a little bit of a stretch. They're not activated until they hit the stomach, and we don't have a lot of fat breakdown really until the small intestine. So it's just important to remember that lipids require a pre-treatment step called emulsification, and emulsification uses bile salts from the gallbladder, and that the gallbladder was just storing the bile and the liver made the bile. Next, we're going to look at the digestion of nucleic acids, which will be very short. A nuclease is an enzyme that breaks down nucleic acids. So we have pancreatic nuclease. There's other nucleases like nucleosidases, but just know that nucleases break down nucleic acids, and there are some brush border breakdown enzymes. And the small intestine is where we begin and end nucleic acid breakdown. We break down nucleic acids to nucleotides, and nucleotides are made up of pentose sugars, nitrogen-containing bases, and phosphate ions. Absorption is another process of digestion, and all food and 80% of electrolytes and most water, so pretty much everything, is absorbed in the small intestine. Most of it is absorbed before the food even gets to the ileum, which is the last part of the small intestine. But the ileum does reclaim bile salts, so they can be recycled and used again for emulsification. Most of the substances absorbed in the small intestine are absorbed via active transport, meaning the use of ATP is required, and they're absorbed into the blood. An exception is lipids, which is absorbed into lacteals instead of blood capillaries. There is some water absorbed in the large intestine, but most of that water is going to be absorbed in the small intestine. And last but not least, here is an overview chart of the digestion processes that include the digestion of carbohydrates, proteins, and lipids. Nucleic acids are left out because their breakdown is very simple. It begins and ends with nucleases in the small intestine. So here this chart shows you the enzyme area that it's secreted into, the gland the enzyme's coming from, and then the enzyme name. We're also seeing the polymer we start with and the polymer breakdown product that we're going to produce. In some situations, this is going to be a monomer because it'll be the end of that polymer's breakdown. So we'll just go through this quickly. It should be review. So salivary amylase is an enzyme produced by the salivary glands and secreted into the mouth. It's going to help us break down polymers like starch and polysaccharides. So that polymer is a carbohydrate. It's going to break them down into disaccharides, which isn't the monomer yet. Carbohydrate breakdown will continue in the small intestine. Those disaccharides will then be broken down into monosaccharides. Breaking disaccharides down into monosaccharides require the brush border enzymes sucrase, lactase, and maltase, and those come from or are activated by the pancreas, and they are going to be secreted into the small intestine. Next, we can look at proteins. So protein digestion begins in the stomach when the gastric glands secrete pepsin, which is secreted and then changed into pepsinogen because of the acidic environment of the stomach. So pepsin is secreted by chief cells, and that pepsinogen, once it's activated, is going to be able to break down large proteins into smaller proteins, and then will continue in the small intestine this protein breakdown into amino acids. Trypsin is a major enzyme secreted from the pancreas into the small intestine that breaks down protein. So be familiar with trypsin. And then last but not least, the small intestine receives lipases from the pancreas, specifically the acinar cells of the pancreas, and that breaks down triglycerides, which is a lipid, into its monomers fatty acid and glycerol. The complete breakdown process would include bile salts, and that pre-treatment step was called emulsification. And that is it for your digestive system overview. Make sure to go back and look at the images, be very familiar with the anatomy, and know the general mucosa and enzyme function of the digestive system.